Welcome to Quantum Photonics. We are here with this very exciting topic from the study of our special guest speaker, and it's drawing inspiration from the inquisitive nature of children and the remarkable adaptability of the brain. Our researchers have embarked on a quest to decode the enigmatic processes behind surprise processing. Using young mice as the subjects, they delved into the realms of auditory perception, exploring how the brain navigates and adapts to unexpected stimuli. Peekaboo, a timeless game cherished by infants, serves as a poignant reminder of the joyous reactions elicited by surprises. Yet beyond the realms of play lies a profound cognitive ability, the recognition of the unexpected, a skill vital for survival in a world where novelty can sometimes signify danger. Through meticulously designed experiments utilizing sequences of sound, they employ the oddball paradigm to uncover the subtle nuances of surprise processing within the developing brain. Their journey takes us from the intricate pathways of the auditory nerve to the cerebral cortex itself, unraveling the gradual maturation of surprise responses from different brain regions. But their discoveries do not end there. They unveil the pivotal role of experience in sculpting the developing brain's response to surprises. Just as a sculptor molds clay to create a masterpiece, the brain forms an internal model of the world, constantly comparing external stimuli to this blueprint. It is through experiences that this model takes shape, allowing the brain to distinguish between the familiar and the unexpected. So, our guest speaker here is Dr. Tanya Rinaldi Barkat. Uh, Tanya is an associate professor in neurophysiology at the Department of Biomedicine at Basel University. Her research aims at understanding the role of specific neuronal circuits in making sense of the sound. She uses electrophysiology, imaging, optogenetic, and behavioral assays to study the development and function of the central auditory system. Prior to establishing her lab at Basel University, Tanya was assistant professor at the Copenhagen University. Before this, she conducted postdoctoral research within Professor Takao Hensch's lab at Harvard University. She completed her PhD in Professor Henry Markram's lab at the EPFL Switzerland. In her career, Tanya was awarded, among other grants, an ERC Standing Grant and Lundbeck Foundation Fellowship. Tanya is a member of the Harvard Society of Fellows since 2008, and in 2007, she received the Corafas Foundation Prize for Outstanding PhD Thesis. She holds a Master of Science in Chemistry from EPFL, Switzerland, and Tanya is a mother to two daughters born in 2010 and 2012. So, without further ado, I bring to you our very special guest speaker, Dr. Tanya Rinaldi Barkat. Tanya, welcome to Quantum Photonics.
Thank you very much, Cecile, and thank you for the nice introduction. Can you hear me well?
Yeah, I can hear
Yeah,
you
it's all
well.
okay,
Can you
perfect.
hear Bob?
Yes, it's all good. Yes. So I think you summarized very well the research and the paper that we came out with. And I will go through more details and explaining, in different aspects of the research. But I think the summary you gave was excellent.
Uh,
So
Thank you. I just want to mention that I'm a fan of Dr. Markram.
Okay, good. Yeah, I learned everything from him. As you just mentioned, I did a master in chemistry. So when I started my PhD in his lab, I didn't have a big background in neuroscience, and I learned a lot from him. So he's definitely a very inspiring person. Actually, with this, maybe I can start by telling a little more about where I come from research-wise and why we got into this question of how surprise changes across adolescence. So as you mentioned, yeah, I did my PhD in Henry Markram's lab, and there I worked on an animal model of autism, trying to understand neuronal differences, abnormalities in a rat model of autism. And I found this subject super interesting, but actually also quite complex. And as you're all aware of, autism spectrum disorder is something that manifests during childhood or adolescence. So I thought actually our knowledge of the normal developing brain without any abnormalities during that period is really poorly understood. So that's why after these PhD studies, I thought, oh, I have to understand better how the developing or what's going on in a developing brain, not only to understand, you know, what a child or an adolescent or how a child or an adolescent perceives the world, but also to understand how all the changes that happen during these early ages actually influence the adult brain. So that's how I actually contacted the lab of Takao Hench at Harvard University, because he was working on plasticity, so the changes that are taking place during childhood and adolescence. And he was particularly interested in a concept that's called critical periods for plasticity, which are time windows during childhood and adolescence in which the brain is particularly plastic to any environment input it might receive. And one typical example of this in humans is that, for example, if we're raised in an environment where we're exposed to only one language, then our brain really gets very specialized to deal with all the phonemes that are present in those language. But in another way, it also doesn't really deal with the phonemes that do not have any specific meaning in that language. So it really has this plasticity during a very specific time window to become specialized to whatever it is exposed to. And in humans, this is, for example, shown if child is up in a Japanese environment without any exposure to any Western languages, then that child will find it very difficult as an adult to distinguish between the two sounds la and ra. Whereas for us, it's actually, I mean, for us who have been exposed to Western languages, is actually quite different. But that's because in the Japanese language, la and ra do not represent different phonemes. So actually, the brain doesn't learn or doesn't have the need to actually distinguish those two phonemes, and therefore it doesn't code for it. And we can train as much as we want in an adult, it will always be challenging uh, to then distinguish them. And maybe for us who for me, like who grew up with a Western language, I grew up with French and Danish as mother tongues. If I'm exposed to some phonemes in the Mandarin language, I will never be able to distinguish them. For example, ma and ma, now I pronounce them the same because I can't make the difference between them. But I know one of them means, for example, mother, the other one means horse. They have totally different meanings, but my ears or my brain has not been exposed to those differences in childhood and adolescence, and therefore it's not able to distinguish between them. So that's a very clear example of critical period for plasticity taking place in the auditory system. And I find this type of plasticity extremely fascinating because it's really what molds the brain, what makes each of our brain very unique as an adult. And it's actually what makes us perceive the world the way we do it. So that's what I studied during my postdoctoral studies at Harvard, and then I became an independent professor now at Basel University, and I actually continue to work a little on this, or at least on what is going on uh, through through development with our brain. And I take the auditory system as a model system. First, because I think technically it's a model system that's actually very easy to access. The stimuli we can use are very diverse, very rich, and we can also simplify them extremely much to be able to understand very simple phenomenon. Plus, after I find the auditory system also fascinating because it's related to language, to music, and to lots of things that have a huge meaning to us as humans. So that's why I work on the auditory system. Um, so maybe to come back to this study, yeah, why am I interested in surprise responses? So surprise responses is actually something that's extremely important in almost everything what we do. It really uh, um, triggers on yeah, increases learning. It's also something that composers for music, for example, use to compose music because they use a right amount of repetitive music with sometimes something surprising that then keeps attention or gives attention to the music and keeps the attention of the listener and keeps the emotion by the listener. So it's really something that is very present in our everyday world, in addition to keeping us safe also. Indeed, if we really go to the basic of surprise, if we don't realize that uh, an input is different from what we expect or from what we're used to, then we can in some situation be in a danger. For example, if we cross the road and we didn't see the car and we don't suddenly hear the sound that's not expected, but that's here, then that would cause a danger, of course. So, surprise has been actually studied a lot in the adult brain, and we know a lot about it. But it has, interestingly enough, never been studied, at least not at the neuronal level, in adolescents. And I think that was really a gap of knowledge that was looking into, especially given that this young brain, so in childhood and adolescence, can be changed a lot. So I thought, why not try to look at how surprise responses do change across adolescence? And if we think a little more about it intuitively, so not with a scientific mind, but if we think about how we experience surprise when we're a child or in children and how we do it in adult, we can already realize that there's quite a difference. And here we can take, as you introduced, Cecile, before, the peekaboo game. So this game where we hide our face and then suddenly we show it again. And if you have tried to do this with the babies, you can see that they really have this surprise response. You can see an example of it on the slide number 2 that I have on the PDF, um, where you really have this surprise response as soon as the baby sees the face. And what's interesting is that at this age, you can repeat such a peekaboo game over and over, and always the baby will have a surprising face and will usually laugh at it or really show the surprise. Try to do the same game with an adolescent or with an adult, and you will see the reaction is very different. Maybe there's a surprise the first time, but then that's it. So already there, just intuitively, we see that surprise responses do change with development. But the neuronal basis behind this were totally unknown between those changes. Um, so surprise responses, as I said, has been studied extensively in the adult brain, and not only at the neuronal level, so in animal models like mouse models, like we use in our lab, but also in human cognitive tasks uh, a typical response that has been measured to quantify suppressed response is called MMN, or the mismatch negativity response. That's a response that a standard measure that we do in humans. And usually the protocol we use is the most simple protocol to elicit surprises, what we call an oddball paradigm, which is a continuation of sounds where suddenly the frequency of one of those sounds changes. It sounds something like do 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 do, something like that, where you have one sound that's repeated a lot of times and another sound that sometimes comes in that's a deviant. So the sound that's repeated a lot, it's called the standard tone, and the other one is called the deviant tone. In my example I gave you, the only thing that changes between the standard and the deviant tone is the frequency, and that's what we have used also in our studies, such a simple uh, oddball paradigm. However, it has been shown that this can work with many other things also than just a change in frequency. For example, it has been shown to work also with voices, so something more natural. It has been shown to also give rise to response when instead of the deviant tone there's actually a gap, so no sound at all. It has been shown with white noise also. Deviant responses have also been observed in many other modalities, and the auditory system it's definitely not unique to the auditory system. It can also be seen, for example, in the visual system and other systems too. But it's very easy to measure in the auditory system, and that's why I think it's a very nice model to study this. Um, there are two general phenomenon nowadays that would explain surprise response to an oddball paradigm like this. So one of them is adaptation, and the other is a prediction error. So adaptation to a sound is actually the phenomenon that takes place when a sound is repeated many times. So, for example, in our case, it's a standard tone that's repeated a lot. And if we quantify the response, the neuronal response to this standard tone, we know that the first time it's presented, the response is quite high, but then it decreases with time in what we call adaptation. So that's one of the things that could explain why a deviant response actually is different from a standard response. But another thing is also the prediction error. So that does not rely on the adaptation to a tone, but it's really something that compares in a way what the agent, so whether it's a human or an animal model, is expecting to hear versus what it actually hears. So this is also illustrated in my slide number 2, where there's really the comparison with this of this internal model, so what the agent expects, with the sensory input. And if these two things are not the same, so if the sensory input is different from the internal model, then it gives rise to what we call a prediction error response. And this is what could also explain this surprise response or the increased activity that we can measure when the deviant tone is played. So I will keep talking about those two different mechanisms that are taking place when someone listens to the oddball paradigm, and see which one might play an important role in the development of those suppressed responses across adolescents. Um, there's also a lot to say more about prediction errors and the whole framework of predictive processing, but probably we can discuss this also at the end. So let's now move to the method, to how we actually asked all this. So the first thing I must say is that we did these measures in a mouse model. The reason why we did this in mice and not in humans is because we want to really quantify the neuronal responses, so how different type of neurons respond to this oddball paradigm, and this is not feasible for ethical reason in humans with the resolution we were looking for. So that's why we use a mouse model. It's not because we're particularly interested in understanding how mice deal with suppressed responses, but it's because it allows us to get to the neuronal mechanism behind the neural responses. And it's believed and also partially proven that the auditory system of mice is similar enough to the one of humans that whatever we measure in mice could be actually also what's going on in humans. So we use mice of different ages. So the ages we used were we were starting with mice as young as 20 days of age, that we call P20 mice. We also looked at mice that were 30 days of age, 40 days of age, and finally adult mice that we defined as mice that were 50 days of age or older. Just to make a little parallel with human development, so mice actually do when they are born, they do not hear yet, unlike humans. Humans start to hear 3 months before birth, where actually mice, when they are born, they don't hear anything. Their ears are closed and they only start to hear around 10 days of age. Um, the time when we start to record our mice is 20 days of age, and that's the beginning of adolescence. I would argue it's, of course, difficult to do the parallel with humans, but that's, for example, the age where usually mice are weaned. That means that female, so they are removed from their mothers, they are big enough to be independent and can to feed independently, and brothers and sisters are also separated. So male and females are separated because that's also the age about when they start to be sexually active, and that's also why we argue that's just about the beginning of adolescence. So yeah, we used mice from those 4 different age groups, so P20, P30, P40, and P50, and we recorded the response to this very simple oddball paradigm in 3 regions of the auditory system. So for those of you who are not familiar with the auditory system, it's actually quite a long sensory system compared to other sensory systems like the visual system, and indeed the auditory information goes through at least 6 different nuclei from the ear up to the primary auditory cortex. In the visual system, as a comparison, it's only 3 nuclei. So those 6 nuclei have been studied also in adults as a response to oddball paradigm, and it has been shown that it's only in the 3 last nuclei of this auditory system that an audience suppressed response can be measured. So in a region that's called the inferior colliculus, another called the auditory thalamus, and finally in the primary auditory cortex. So that's why we decided to focus also just on these 3 regions. And the flow of auditory information, it goes first to the inferior colliculus, then to the auditory thalamus, and finally to the auditory cortex. And what's also very important in the auditory system, and probably also quite unique to the auditory system, is that there's a lot of feedback to all of the different nuclei of the auditory system, including the auditory thalamus and the inferior colliculus. And I think I will come back to this later to see how those feedback projections actually play a role in this surprise response. So the way we quantify the surprise response is that we played those oddball paradigms. Again, you can see an illustration of them on the slide 3. So it's the example I gave you before, a standard tone, which is sometimes replaced by a deviant tone, where the two just have a difference in frequency. We then also play this tone parallel so that there's no bias to one particular frequency, because we don't really care about the frequency. We just want to have one that's deviant and one that's standard. And then we quantify the responses of individual neurons to those 2 tones, so the standard tone and the deviant tone. And in general, the response to the deviant tone is much larger than the response to the standard tone. The way we quantify this is with our an electrode that we insert into those 3 regions of interest. With this electrode, we can actually measure the change in voltage that gives us a direct measure of the spiking activity of the neurons. We can then quantify how many spikes those neurons make as a function of the different sounds they play, and then we can quantify the amount of spikes that are generated as a response to the standard or to the deviant tone. We then quantify these responses in what we call SI, or the surprise index, which is a response of the deviant tone minus the response of the standard tone divided by the sum of both responses. And if this number is positive, that means that the response to the deviant tone is larger than the response to the standard tone. And again, to get rid of any bias that could arise due to the choice that we make of the frequency of those 2 tones, we actually use what we call a combined surprise index, or CSI. That's the sum of the deviant responses of both frequencies minus the sum of the response of the standard frequencies divided by the sum of all of them. And here again, if this number is positive, that means that the tones give rise to larger responses when they are in deviant context than when they are in the standard context. And then we've quantified this across our 4 ages of interest as well as the 4 regions of interest. And the results are summarized in the slide number 4. So what we observed first generally was that the surprise response is smaller in the inferior colliculus, gets larger in the auditory thalamus, or MGB for medial geniculate body. That's a technical name of the auditory thalamus, and that it gets even larger in the primary auditory cortex, or A1. So this was not surprising. Indeed, it has been shown in the adult brain that the surprise response gets larger the higher up we come in the auditory system, and we could replicate this not only in adult mice but also in younger mice. However, another result we obtained was extremely surprising and not what we expected. That's how it changes with age. So in the inferior colliculus, we actually did not see any change with age. The surprise response was as small for all ages from P20 to adulthood. However, in the auditory thalamus, MGB, the response to surprise was much larger in P20 old mice and then decreased significantly at P30 and then stayed stable until adulthood. So there was a decrease from P20 to P30 in the suppressed response in the auditory thalamus. In the auditory cortex, there was also a decrease, but interestingly, it happened actually at a much later stage, and there it's only between P40 and P50 that we saw a significant decrease in the surprise response. So the surprise response was quite large at P20, P30, P40, and then it decreased for adulthood. So this results was extremely interestingly and surprising. It showed that deviance response matures sequentially along the central auditory system. So it seems to be mature first in the auditory thalamus, in the inferior colliculus, then in the auditory thalamus, and finally in the primary auditory cortex. And this maturation is not an increase as we had initially thought, but actually a decrease in the standard response. So the next step in the study was to try to understand how this decrease in surprise response could arise. What could be the reason for such a decrease, and why would that decrease be different in the different brain regions we looked at? So we did quite a lot of controls to look at this. We, for example, looked at the cell properties across development to see if the neuronal intrinsic properties or response to just standard tones change across development if the tones were presented by themselves. And we actually did not see any changes in the neuronal properties of individual cells that could explain the change in the suppressed response, meaning that those suppressed responses were not due to maturation of the cells themselves, but it was really something related to the surprise, so to the neurons put in the context of a oddball paradigm. For example, the threshold of response of the neurons didn't change. The number of frequencies they were responding to, what we call the bandwidth, did also not change. Their spontaneous baseline firing activity also did not change after 20 days of age, and this in none of the regions we studied. Another question we were interested in was to understand whether these changes were more related to the adaptation to the standard response or rather to the prediction error to the deviant response. So was this change more related to the change in standard or more to the change in the deviance response? And here again, we did a lot of controls, and I don't want to go into details about it now. But what we showed is that actually adaptation cannot explain the change in suppressed response we observed. Therefore, we think that those changes we described are more related to a change in predictive processing or in the prediction error uh, that matures will develop. Um, Another question we ask is what was the role of the auditory environment on these changes? So in order to address this, we wanted to try to raise mice that were not exposed to any, that did not receive any specific auditory input during the time period we were interested in, and see if that would change anything in the way they were dealing with surprise responses. So this is an experiment that's actually quite easy to do. For example, in the visual system, where to remove any visual input, we can just suture the eye of mice and then prevent any visual input to reach the eye. Such a type of experiment is slightly more challenging in the auditory system because indeed, even if we close the ear of the mice and prevent sounds to enter the ear and the auditory information can always go through the bone conduction and therefore still stimulate the auditory system. So if we just close the ear of mice, this will not prevent them from hearing something. So we use a trick that has been used also by many other labs to try to uh, remove auditory input. And for this, we actually don't remove it, but we mask it with a continuous white noise. Uh, White noise is something that you, for example, here, if a radio is not working very well, that never happens nowadays, but it used to. It's something that sounds like shh or something like that. And that actually covers any a lot of the structural sound that one hears in the auditory system. And it has been shown by us in previous studies, but also by other groups that this is actually equivalent to removing structured auditory input. So we used this trick and we exposed the mice to this continuous white noise from the age of P20 when we start recording them until P50, until adulthood. And then we looked at their responses to the surprise, to the deviant tone when they were adults. So as soon as we took them out of this white noise exposures and we recorded both in the auditory thalamus and in the auditory cortex to see if this removal of auditory environment would affect the surprise response. So the first results we obtained was in the thalamus. You can see the results on slide number 5, where we showed that actually this white noise exposure had no consequence at all on the suppressed response, and this was still exactly the same in P50 mice when we compared control mice and those that had been exposed to white noise. So it was still smaller, significantly smaller than it was at P20. However, that was not the case when we measured the deviant response in the auditory cortex, whereas there we saw a significant difference between P50 control and white noise exposed mice, whereas the white noise exposed mice had actually a deviance response that was now the same as they had it at P40 or P30 and P20. So it was like if this white noise exposure actually prevented the maturation of this surprise response and kept it in this immature state. And what was really interesting here, what this result allowed us to conclude is that the auditory environment really influences strongly the surprise response. And if we raised in an auditory environment, an abnormal auditory environment, or without a proper hearing, then we can really have an abnormal response to surprise as adults. Another most interesting conclusion we could do from this experiment is that the plasticity here that we observed was taking place in the cortex only and not in the auditory thalamus. So it seems that this plasticity that is taking place in adolescence is really of cortical origin. And if we think about other studies looking at plasticity during adolescence, like the studies on critical periods, we know that this plasticity is also unique to the cortex. So it seems that during adolescence, it's really the cortex that changes and that allows the brain to actually adapt to its environment and in a very adaptive or maladaptive way. And it's nothing that's changing in the subcortical circuits. So that was a very interesting observation we did there. So we were still not satisfied with these results, and we still wanted to have a better understanding of what kind of circuits could be related to this maturation of surprise response across adolescence. Now we focused mainly on cortical circuits because we showed that the plasticity here was particularly interesting in the cortex rather than in the inferior colliculus or in the thalamus. And one of the first things we did was to look at the different cell types that were involved in this, and we compared the main inhibitory neuron cell type, which are called fast spiking neurons, which are putative parvalbumin neurons, for those of you who know what this is, with excitatory neurons. So excitatory and inhibitory neurons are the two main type of neurons, or the two types of neurons in the brain. They can be subdivided in many other cell types, but we very um, roughly looked at the main type of inhibitory neurons and compared it to the response of the main type of excitatory neurons, and we could actually not see any difference between those cell types, meaning that this suppressed response is probably something that's coded by both inhibitory and excitatory neurons. We then also wanted to see where in the auditory cortex this particular type of plasticity was taking place. And for this, we looked at the different layers that form the cortex. So as some of you might know also, the sensory cortices in all mammals can be divided in different layers. And there are three types. There are 6 layers, but they can be divided in 3 types. The input layers, that's the layers that receive the input from subcortical regions. So the superficial layers that then receive again input from other regions, but also from these um, input layers. And then there's a deep layers, layer 5 and layer 6, that are known to then send information to other brain regions. And thanks to an analytical trick, we were able to actually separate the uh, measurements we did in input, superficial or deep layers. The analysis we used was called is called current source density analysis, and I'm also happy to go into details of this if needed later on. That's represented in slide 6 of my presentation. And we could show here that it was actually particularly the deep layers of the cortex that showed this significant change in suppressive response across adolescence and none of the other layers. So this analysis allowed us to really focus on the neurons that were in those deep layers of the cortex as the ones that actually are able to really code for the change in surprise response across adolescence. As I just said, those deep layers are important because they do send information to other brain regions, and one of the regions that they send their information to is back to subcortical regions. So it's known that those deep layers send information back to the auditory thalamus and also back to the inferior colliculus, among other regions that they also innervate. So we were particularly interested in those connections back to the auditory system, those that we call corticofugal projections, to see if they would probably be responsible for the changes we saw in response in a surprise response across adolescents in the auditory thalamus. So in order to look at their role, we used a technique called optogenetics that allows, with the help of light and genetic manipulation, to control a specific type of neurons by just shining light on them and either excite them or inhibit them. And to cut a long story short here, we were able to silence all projection from the cortex to the thalamus and to show that this change actually removed the developmental changes that we had observed in the auditory thalamus. So this feedback connection from the cortex to the thalamus are probably responsible for the developmental changes that we observed in the auditory thalamus. And we did a few different controls to show that this was specific to the corticothalamic connections, and indeed the connection sending the information back to the inferior colliculus, what we call the corticocollicular connections, were not affected, or were not affecting the deviance or the suppressed response in the inferior colliculus. So it seems that it's really the connection back to the auditory thalamus that were changing across adolescence that explained some of the changes we observed in the auditory thalamus uh, with this maturation across adolescence. So to summarize the study, maybe you can go to slide 8 now. I have done a schematic of this, so it seems that deviance detection is changing, or suppressed response is changing across juvenile development, and particularly in the auditory cortex, where it's the deep layers of the auditory cortex that seem to be changing particularly much between early adolescence until adulthood. Interestingly, those deep layers also influence the feedback connections to the auditory thalamus, and that might also explain some of the changes that we observed in the auditory thalamus. In addition to this report of the observation we did with the results, those result allows us to really pinpoint to a specific role of the cortex. Indeed, the cortex allows the maturation of suppressed response well into adolescence. Without the cortex, these responses would not change so much. And it's really in the cortex that it matures up to adulthood, actually. The cortex also has this very specific role in the plasticity. It allows experience-dependent change in deviance detection. Indeed, when we manipulate the auditory environment for those mice, we could see changes in the cortex only and not in the auditory thalamus. And finally, we also showed that it might be the many circuits that are involved in the change in suppressed response across development, that they are all located in the cortex, in the deep layers, but also in the connections that project from deep layers to other brain regions. And in our particular case, we looked at the projection from the cortex to the auditory thalamus. So that's a summary of our results. Maybe now I can just go to the last slide, or slide number 9. To see why actually it's interesting and what could be the implication of the type of results we have obtained. So one of them is, of course, to better understand the brain and how it develops, to better understand how we perceive not only deviant sounds or surprising sounds, but also how we perceive in general. But another application is for the case of when the brain is perceiving the environment in an abnormal way, and particularly for developmental disorders like schizophrenia and autism spectrum disorder. Indeed, actually, in schizophrenia, it has been shown that the suppressed response, now quantified as a mismatch negativity response, is significantly different in people suffering from schizophrenia as compared to control, to the point that actually this mismatch negativity response is now almost used as a biomarker for this disease. Um, so we don't really know yet how it's different or why it's different in schizophrenia, but that's now that we have a better understanding of the mechanism of what circuits could be involved in response to surprise and how it could not develop properly across adolescence, we can actually try to think about it in the context of schizophrenia and see what could be at the origin, what circuits could be malfunctioning or abnormal in the case of schizophrenia. And we can make the same conclusion for autism spectrum disorders. Indeed, it has also been shown in some cases of autism that the mismatch negativity response is abnormal in those patients, and therefore it could also give some hint of what type of connections, of what type of neuronal maturation could be something we would be more interested in to try to understand this condition a little wider. Um, yeah, I think I will stop here. I mean, yeah, maybe just to summarize the perspectives that this our study could give is that I repeat myself here, but that the refinement of surprise response is mainly a cortical effect, emphasizing the cortex as being a highly flexible and adaptive system, and what really allows each of our brain to be unique. Another perspective is also that predictive processing can actually be manipulated during juvenile development, and I think that's also super interesting that we can modify and influence or change or maybe restore maladaptive predictive processing, and that's particularly the case for disorders. And indeed, the circuits implicated in this surprise response suggest also possible targeted interventions to treat neurodevelopmental disorders like schizophrenia or autism, but also neurodegenerative disorders. Indeed, it has been shown also, for example, in Parkinson's, that the mismatch negativity response is in some cases abnormal. Yeah, and before opening the floor for two questions, I just want to thank also all the people involved, and you can see on slide 10 the whole group. And I must be particularly thankful to Patricia Valerio, who is now not in our lab anymore, but who has really been the driving force behind this project. She got the help also from Julien Reichenbach, Suyash Yoshi, and Gioia De Franceschi, that were all former postdoc in the lab for the analysis part of the results. And I thank you for your attention and look forward to your questions.
Yeah, thank you so much, Tanya. We would like to inform the floor that we are opening it, and for those who would like to speak, to ask questions, please feel free to raise your hand. We will bring you up the stage. And I would just like to reintroduce our guest speaker here for.
Hello? Can you hear me?
I
Okay.
can I can hear you.
Oh, okay. Okay. Sorry, Cecile. I misunderstood. My question was kind of pertaining to the diagnostic nature. You kind of hinted at that just a little bit at the end there, that perhaps, it could be something like perhaps some gadget future. We have something like a acoustic panel or auditory panel, or I don't know what you would call it, maybe an inventory, uh, like an adult inventory. This would be like an adult inventory, auditory acoustic monitoring, something like that. That's kind of what I'm thinking in terms of the phrasing or describing it to somebody else who wasn't here at the talk. But do you envision that being something that gets developed sometime in the future? Like, I know Apple had something where they were tracking the eye movement of children during a tracking task. and
Hmm.
that allowed them to make diagnoses of autism. And this seems to be a similar. This seems like the acoustic analog of that a little bit. So if you had any comments on that, or if you were familiar, thanks.
Yes. Yeah. Yeah. Right. Actually, so this mismatch negativity response, which is actually the equivalent to the surprise response, but measured with electrode that are on the surface of the brain, not inside the brain, is actually used in some cases as a biomarker, so really as an analytical tool. And I think that's quite early days for this, because it's surprising that, I mean, it's good to call it surprise, but it wasn't expected that the surprise response was different. But now it has been shown in so many patients, and it's so consistent across patients that actually it could be used as a biomarker. And that's super useful, because if we, I mean, I don't think we have any other convincing biomarkers of schizophrenia, for sure not of autism, um, and that would allow to have more of an objective way of quantifying it, so without relying on the report of the subject, and that would be a huge advantage. And also another advantage of this is that it could probably be also um, um, looked at earlier on in the disease or in the conditions, before actually the symptoms become too important, and maybe there could be some prevention. So biomarkers for any type of conditions are extremely important, and they are lacking for autism for sure. I think for schizophrenia there's also nothing really convincing. We can't measure anything in the blood or anything like that. So if it could be used, if this mismatched negativity response, the suppressed response, could be used as a biomarker, that would be fantastic for many reasons. So I think the field is going in that way. I'm not a clinician, so I can't really, one would have to talk to a clinician, but I think if we can go in that way, that would be fantastic. From a scientific point of view, if that's true, this actually really allows us to pose hypotheses about what's lying behind those conditions, what brain mechanisms are different under those disorders. And that's also extremely important to try to tackle these disorders with something different than, for example, just an abnormal gene or things like that, which is most commonly used nowadays to try to understand better those disorders. So for sure, I think it has a huge potential as a diagnostic tool, both for research, but of course also for patients.
Okay, so the final question, and this is kind of a out of left field question. So some of the work that I'm doing with folks out of UBC, they're looking at experiments, or University of British Columbia here in Canada, and they're looking at
Mhm.
experiments where maybe there's some sounds that, not like as you say, surprise, but there's also like when people have. Um, like maladapted coping experience sometimes have positive indications of therapeutic I don't know, tricks that people come up with. So some people go doo 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 or some other kind of sound, and there's something that they're able to do in this kind of, and it feels like the diagnostic question. So I work in diagnostics, specifically viruses, and I work with a few other labs on some other project. And this project in particular is delving into this weird biomarker. So for me, it's a very strange thing. It feels very much like a cloud that we're touching when anyone says biomarker. So
Mm,
I find it a little bit uneasy, but
yeah. Yeah, it could be. I mean, ideally, biomark is something you measure in the blood with a machine and it gives you a number. But in this case, actually, mismatch negativity response or spread response is also observed, for example, in anesthetized mice. So where the subjectivity is not there. So if it's a neuronal response, then it could be actually as it could then really be a biomarker in the sense that it's something totally objective and not dependent on the on the person's state or report or something like that. And
but
I
you
think there
but
it
you
would
made
be
a
really powerful.
yeah, but you made a comment at the beginning. I thought it was very poignant that the 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 cultural effect of auditory tuning
Yeah.
perhaps causes someone to feel that something is unexpected, but actually it's very expected in this other language. So you just are not familiar, and so
Yeah.
that was an interesting bias that you worked through. So, I guess final question is, where do you see this work going, or I guess you might expand to other processes other than surprise, or
Yeah. I mean, we're studying in the lab a lot of other plasticity phenomena taking place during development related to those critical periods, as you said. We, for example, look also at how, how we can categorize sounds as belonging to the same class or category. Um, and that can also be related to surprise, by the way. But for example, when I say hello or when someone else say hello, the frequencies used will be different, the intensity will be different, but somehow our brain also is able to categorize this as something having the same meaning. So we're looking into how this changes across development, and I think that will also tell quite a lot about how the brain processes things. To come back to developmental neurological diseases, it's known in some patients are suffering from autism that categorization is very difficult. For example, I report from Temple Grandin, a famous autistic person who actually wrote a book about this. She said that for her it was very difficult to say that to distinguish a cat from a dog and to say, oh, this is a dog or this is a cat when she then looked at a new dog. So for her, it was very difficult to categorize things. For example, to see that one street was different from another street. Or, I mean, categorization in general was very difficult. And I think that's something we can really address very easily in the auditory system. And we're now looking at it through development. We are in terms of predictive processing or in surprise response, we're actually also looking at paradigms that are slightly more complex than this oddball paradigm. So the oddball paradigm we've been using is the most simple that can at all exist. It's really just a change in frequency, but you can do it much more complex. And with this, you might actually you will trigger different neural networks and circuits. And then we want to see also how those changes with development.
Oh, okay. I guess something that was surprising for us was that we were able to see something like drift with multi-sensory integration. Was there
Mhm.
anything like that on your end as the animals got adapted to the surprises? Because I guess you can only surprise them so much, so it's kind of a difficult experimentally.
Yeah, so we've tried to address this, perceptually, how to try to quantify how they feel the surprise. What we have quantified is only the response to the neurons. They decrease with age. That's the only thing we can say. It's not with for us with I mean, we didn't want to see if they learn the meaning of the sound, if that influence, because then the sound have a meaning, a saliency, and then it's the notion of surprise, and it's more difficult to talk about. So we haven't looked at that yet, but yeah, it's not that easy to address. I mean, ideally,
Well,
we would want,
if you did something like instead of an unexpected tone, you did like a predatory bird call.
yeah,
That's something that, for example, caused epigenetic changes in another
yeah,
study, and
yeah.
that I
This
think
would be
speaks to some level of encoding, right? I think
yeah,
uh, inevitably, inescapably.
yeah. And also, we could use mouse calls. For example, mouse calls like the standard would be another pup call, and then suddenly a male call, an adult male call, and see how they react to this. For sure, there are many possibilities we could look at it also. Yeah, step by
Cool,
step.
thank you.
It's good ideas. I think we should definitely, and we're definitely not done with it.
Yeah, thank you. Thank you, Eric, and yeah, Tanya. So, yeah, if there are other people who would like to come up the stage to ask questions, yeah, please raise your hand and we will bring you up the stage. So, that also made me think the element of surprise. I was thinking about the implication of your study on how one makes a successful summer song. So, of course,
Mhm.
usually every summer in the US, we try to make a summer song, and some people try to call several musicians who make certain processes, and then in the end, the artist, tries to choose a summer song, and then there is some kind of a formula that makes a summer song successful, meaning to say maybe it also is affected by the surprise factor and also the prediction that's happening. What can you say about this?
Yeah, I think that's also an extremely interesting direction to go. So, um, o- obviously, we started with the most simple oddball paradigm that cannot address your question, but that would be also one of the next step to go. As we know, when we do music, what we like in music is that some things are expected, that you have at rhythm. If you think about music, there's a lot of repetition, but then sometimes there's also something unexpected, and it's really this mix of what is expected, repetitive, with and what is not expected, that gives a tension, and that makes that we actually enjoy the music and want to listen and makes the music interesting. And if you want to read more about it, I would really strongly recommend a book by David Huron called Sweet Anticipation, Music and the Psychology of Expectation, um, where yeah, it really puts forward actually the advantage of surprise, and not only the disadvantage. When we think about this, we're like, oh, maybe, surprise, we don't like it so much if suddenly there's an unexpected sound and we startle. But actually it has a lot of positive things, and has been proven by a lot of studies that surprise has important roles, for example, learning, exploration, for memory formation, also memory segmentation, and so on. So yeah, I think for your next song on a global way, it would be good if you have the good mix of something people can relate to, so something expected and familiar, but with also a good mix of something unexpected that keeps this tension, and that keeps a surprise.
Yeah, thank you so much. That's why some parts of a song are repeated, so that in a way, when you try to, after you've heard the song, it becomes you have an earwig and it repeats
E-
on your mind.
exactly. And if you compare this with language, for example, a song is much more repetitive in general. But that's also what we like in a way. Yeah, that then we can also repeat the melody and things like that. But then sometimes it's a little different, and I mean it has to have a good mix of both, I think. But it's definitely the repetitive aspect of a song is extremely important, and probably
Sorry,
even
can I
more
just?
in kids.
Yeah, sorry, I just wanted to ask what was the name of that author so I can post a link to the book.
Yeah, David Huron. Huron, it's Huron.
HUROOL.
O-N. N. Maybe can I write something?
Yeah, did you get it, Eric?
I'm just checking right now because I might actually buy it because my wife is a mathematician who studies music, and one of the side little projects that we have is actually a game that focuses very heavily in VR on sound design, and so keeping these kinds of ideas, uh, kind of oddly, not to too much of a pun, but resonate with us. So
Yeah,
just wanted to get the source down.
and there are few formulas in this book, so I think a mathematical background will be definitely nice to have. It's called Sweet Anticipation, David Huron. It's a little more psychological than neurological, but I think the idea of, I mean, that's the whole idea of predictive coding also, why predictive coding and suppressed response is also an error in predictive coding, and this might be one of the mm, basic factor or basic property of how our brain works. I would say I think it can be really, if we look at our brain or whatever we do with it in the context of predictive processing and how surprise is dealt with, I think it can really explain a lot of things, including how the brain learns, how it develops, and so on, and how it makes music or enjoys music.
Yeah, I think that's what gives a feeling of pleasure to those who are doing just improvisations, because of course you are anticipating something, but you are not sure, and there's an element of surprise there when the musicians are trying to jump doing this just improv, because your playing will never be the same at all at any time. So I think that's what's going on, and in the flow that they have during this experience, there is always an element of surprise. Yeah, so
Yes.
I'm
Yeah.
just reminded about it. Yeah, so at this point, we see Kevin coming up the stage. And yeah, Kevin, if you have some question, you're welcome.
Yeah, I came in late, but and I'm still waking up. But it reminds me of as well as my question. If I got this right, I know this is obviously a mouse model like normal, but for humans, I heard you speak of anesthesia where the mouse brain still reacts and it's to stimuli, I believe. But
Mhm.
so for drugs, alcohol, drugs, caffeine, tobacco, what have you.
Mhm.
Do you think that may reset, these results, and change them and disrupt them, and one's, just typical things in psychology, like, one's mood, just like me, early morning,
Yeah.
meditation, stress, work, it's stuff like that. What
Yeah.
might affect this?
Yeah. So for this, I have to maybe take a step back and say that there might be different type of surprises actually. So we are looking at the most simple one that's also repeated in anesthesia state. For this one, I think it's probably, you know, the brain state in which we are does not influence it too much. So depending on, it might influence a little, but as you say, drug or caffeine, alcohol or something. I think the surprise response is odd. Yeah, this oddball responses I looked at in this study might not be affected too much. However, some other surprises that can be related to some other mechanism are strongly affected by this. And in the field, we talk about two type of deviance. So the local deviance, that's what I've been looking at, is that when you have something standard and then something deviates from it. And then you also have the global deviance. For example, if you're used to have a melody, and this melody can have different frequencies, and you repeat this melody a few times, and suddenly one note in this melody is changing, and this is called a global deviance. So, and this might rely more on a different type of working memory. Again, we don't know exactly what it could be, but this type of surprise, more global, are definitely affected by the brain state. And actually, I've also heard that that's used as a way of diagnosis for knowing if people are conscious or unconscious, if they have this global response. So the local response, the one we have been studied, is not affected too much by different brain states. So global response is strongly affected by it. And I would expect that this type of surprise response is definitely affected if if you are tired, if you have drunk alcohol or medication and so on. This will definitely be affected by it. It's not addressed directly in our studies. That's also on our to-do list of what could be addressed and what would be super interesting to address in the future. And it might be different circuits than those that I presented here that might be involved. I wouldn't be surprised that other mechanism are then involved on top of the one we looked at for those more global surprise responses.
The oddball response. Okay, yeah, I can't wait to look into all this. Thanks a lot.
Yeah, you're welcome.
Yeah, thank you, Kevin. So I wonder if Bob is able to speak now to read to read the latest question of Geary on the room chat. Bob, are you able to speak?
Yes, yes, Cecile, thank you very much. Geary asks, on a processing level, how are surprise, alertness, excitement, and fear, how are they the same and how are they different?
That's a very good question. Can you, so you said fear, surprise, excitement,
And
or
surprise, alertness,
alertness?
excitement, and fear, how are they
Yeah,
similar and how are they different?
yeah. So that's a very good question. And here I think I have to say that there are very different types of surprises, and I think those like fear, excitement, alertness. Alertness, it's a different state probably because we can be alert without. Maybe what triggers alertness is maybe a surprise, but then we can stay in an alert state for a long time. But I think surprise goes up and down. Um, Fear and excitement, I think, are two different aspects of surprise, and I think there also surprise can be we kind of in some uh, some of the previous research on surprise in the field has been quantifying surprise as one single thing, and including our story. We just looked at oddball and we called it surprise, and that's it. It was very simple, but probably too simple. And I think there are different type of surprises, and here I can also point to some papers that actually use modeling to try to quantify the different type of surprise. And in these papers, they, for example, talk about a prediction surprise. So I think that's more similar to what we've been looking at, something that doesn't fit with uh, with what was expected. But there's also change point detection surprise, confidence corrected surprise, information gain surprise. I think that would probably be related, for example, to excitement. That's when we see something new and we learn from it and we're excited by it. I think that's also another type of surprise. Fear surprise, I would say that's more like a protective surprise, what we need when we have, for example, to avoid the car on the street or something like that. Um, I think it's all different aspects of surprise that probably all start with something similar to the oddball paradigm surprise that I have been describing in this paper, but that I have other aspects on top of it. And for example, fear definitely has emotional aspect of it. The amygdala, which is one nucleus in the brain that's very much involved with emotion, would definitely play a very important role in fear response. And yeah, it is definitely a surprise response. At least as the first time we hear it. After it would be probably something different than surprise when um, when we, fear is sometimes associated with something new. But maybe if a mouse listens to a cat a few times and the tenth time it hears a cat coming, it's not a surprise anymore, but it can still trigger fear. So it can also be something different in a way. But there's definitely different quantification of surprise, and I think to better understand it, we scientists should be more clear about what aspect of surprise we're looking at and to try to understand it more precisely.
Ah,
I
thank
think,
you. Bob, did you
uh,
notice Giri is here on stage? He might have a follow-up question because he was the one who asked it. So we'd
Roger,
like to Giri if he has a follow-up question or comment.
Thank you, Cicely. But I think if at all Bob or anybody else have anything to add on to it, I would listen to it first, please.
No, we're
All
saying
right.
you should just you can just present your question. We're all excited to hear it.
Okay, I'll clarify this. There will be some similarities in the how these things work, how it is processed,
Mhm,
but the outcome will be different. I do understand that,
mhm,
but there will be
mhm,
some similarities also, and there will be differences. Like for example, let's say how we perceive different speech sounds.
mhm,
If we are like there are similarities between vowels, how it is processed, and consonants, but it is
mhm,
entirely different also. And consonants in within themselves there are similarities, and within themselves there are differences also. So probably these four emotions which I just stated will have a level of similarity in which they are processed, how we process it inside. And definitely the
Mhm.
outcome is different. It is perceived as different only, but the
Mm.
processing to some extent it could be same. And I was just wondering what is your understanding regarding that. Like it will make it very easy for me to understand also if you already have a good view about it.
Yeah.
That's why. So
So I fully agree with you, and I like that you actually pointed out the fact that the four emotions that you mentioned are emotions. And what we looked at here is a very small regions of the brain. So it was three regions, but they're still all very much related to the auditory system, to the sensory processing. And they may be very much related to what changes in the sensory inputs of frequency or things like that in our case. However, this information is after sent to many other regions, and including, for example, the amygdala that deals with emotions. And the amygdala is known to receive information from the cortex, but also from the auditory thalamus. So I would be also very interested to do exactly the same study we did, but instead of putting our electrode in the auditory system, put it in the amygdala and see what's going on there. And there we could, do the measure. Now, with the oddball we took, we on purpose didn't want any saliency to the sounds we had, so they had no meaning. We didn't want any emotion related to this because we want to start at the most simple level where there's only a sensation, so an auditory input. The next step could then go to see what if we give a saliency to this sound, so something that could trigger excitement or fear. How would it look like in the auditory cortex? In the auditory cortex, it might be that it doesn't change so much, or maybe it does, but maybe not. But I'm sure that if we look, for example, in the amygdala, the region that deals with emotion, or maybe in motor systems also that deal with the consequence of an emotion. For example, do I have to freeze? Do I have to fly? Do I have to, be happy? And there it might be affected a lot by the meaning of the sounds themselves. And we haven't looked at it yet, but I think that surprise can definitely not only be quantified in the sensory systems as we have done it, to really have an understanding of how we perceive and feel it or how we put emotion on it. We have to definitely look at other brain regions. And they also definitely change with adolescence and development. And if we think about when we were younger, or if we think about our kids, we definitely know that this is changing a lot during this time window and can be shaped in a way and modeled and modified a lot. So that's definitely also interesting questions to go to. On purpose, our study was really as simple as possible so that we can go, step by step and really try to dissect the different mechanisms involved in this type of processing. Also, if we go back to music, for example, what we talked about earlier, music brings emotion. That's why we listen to it. So also to understand what we like or not in music, or what it, then we have to look at other regions and just the sensory processes. Does that make sense?
Tanya, if I may, Tanya, like if at all you are taking an auditory response,
Mhm.
auditory responses generally it is classified into short latency, medium latency, and long latency responses. And
Yeah.
cortical activities are generally seen in long latency response. But
You
if it
think
is
you
the
talk
emotion,
about MMN or things like that
electrical,
now? Yeah,
yes, MMN and things, yes, yes.
yeah, yeah,
A emotional response usually should be coming after the cortical representation, right?
yeah. I
In the
would
sense
guess
like
so. Yeah,
it should be even beyond the long latency responses, right?
yeah,
So how do you isolate it when you're trying to study it?
yeah. So the latencies you're talking about, I think it's those that you measure with in humans with non-invasive electrophysiology. What we study when we put our electrode in the brain, we actually have latencies of about 10 to 15 milliseconds, so super fast. in the auditory thalamus is like 8 to 10 milliseconds. And we can really follow, when we are invasive with electrodes in the brain, we can really follow, where the information is triggered first. So for example, the amygdala with the emotions, it comes early, but it does receive input from the thalamus. So there, we can definitely measure the latency there. In humans, it's less precise because we cannot be that close to the neurons. But I think that's also one of the advantage of looking at animals for this type of question, is that we can really follow where it goes. And I think the emotion comes after, in this. I mean, if the emotion is triggered by a sensory input, it comes after the information has reached the auditory thalamus for sure.
Although
Unless
you
it's
may
a
eventually,
reflex, and that's even earlier.
yeah, eventually you may just get a fleet of people who have a bunch of Neuralink, and then you can do all sorts of experiments with those folks,
Yeah,
get them to comply. Click here to be part
yeah,
of this study.
yeah. I'm not sure if that will be tomorrow, but yeah. I
Well,
mean,
there's already one
more
person
and
who
more,
received it, right?
yeah,
And if
and
you
more and more
and anyone's
studies.
gonna
Sorry.
No, no, I was just going to say, if anyone's going to pull off getting neuroscience as a thing, it's the guy who got electric cars as a thing. And so I think Elon has done
Mm,
quite a lot for neuroscience, making it kind of more of a mainstream thing. So I think it might actually be much sooner, especially given that the recent first patient received Neuralink, I think about a month ago
mm,
now. And they're able to use a mouse cursor, which is crazy. So
yeah,
that's the kind of timing, if they can get that timing down, then that already is quite invasive.
yeah,
But
for sure. Yeah,
quite, if it's equipped in the way that your laboratory needs the components, then you could probably get the time of flight and other things like that. I'm not sure if
yeah,
you're using those kind of
yeah,
parameters, but yeah.
yeah. Actually, there are more and more studies done in humans, and to my knowledge now, it's mainly done in patients. For example, Parkinson patients, they get this deep brain stimulation electrodes to treat the disorder in some cases, and there we can also record the activity. People with epilepsy or stroke sometimes also get electrodes to treat the disorder, find out what is not working, and we do get extremely valuable data from those patients that are not only related to the conditions they have, but also address basic science questions, neuroscience questions, and I think that's extremely promising. And it's really, it's crazy the amount of data we can get now from human patients compared to before. So for sure, it's something I guess we can. I mean, that will be extremely valuable.
Yes, so we have Kyle here who came on stage. And before I give the mic to Kyle, I just want to ask you, Tanya, were you the one who asked me that you are also interested in what Miguel Nicolelis did here
Ah, yeah.
in
Yes.
Yeah. So I just want to tell you that he might be coming back again here soon because he set up a lab in Europe, and it's supposed to be, of course, dealing with non-invasive brain computer interface for
Okay.
therapeutic reasons. So if I would be able to make a schedule with him, because they sent me a message, but I told them I was traveling. I invite you to be my co-moderator here
I would
when
love to.
Miguel is
Yes.
here.
Yes, that would be really nice.
Yeah. So anyway, yeah, I look forward to that. And at this point, I'd like to give the mic to Kyle, who's been patiently waiting. Kyle.
Thank you. I just want to say thank you. It was an amazing presentation. I was just thinking about how a lot of people think of one spectrum, but people such as like Dr. Karen Parker talked about multiple spectrums. So there is autism without intellectual impairment, and it's
Mm-hmm.
often associated with valuable technical and social skills
Mm-hmm.
and heightened perception.
Mm-hmm.
So what I'm wondering here is if there are actually people that are gifted with auditory perception and attention. There have been experiments that show that some people have just a hyper or just a better heightened perception of pitch than others.
Hmm. Hmm.
So I was just wondering if there are people that might be experiencing some of what your research is showing, that are being placed in a camp of autism, but actually have heightened perceptive skills.
Yeah, yeah. I sadly do not have any data for this, but I fully agree with you. I think I worked with a model of autism for my PhD, and I think um, maybe even more and more we put so many things into autism, which I would not be surprised that in the future it will be considered as very different conditions, I mean, not disorder, already now it's called a spectrum more than a disorder. And yeah, I think that if you look at the symptoms or think about different autistic people, they just are so different that I think it's kind of sad that they're all under one umbrella, which might not be the case at all, but probably there's now a better way of doing it now. So again, to come back to the discussion we had before about biomarkers, I think if we can find a biomarker, it would probably help also to separate those different spectrums, um, because we can then, more objectively quantify the different things. And I think what is extremely difficult I mean, probably in any psychiatric disorders, but particularly, I know the case of autism is that it's so vague, the definition, and it is people, every autistic person is very unique. The consequence is also that treatment is extremely difficult to develop because everyone is so different. So I really hope that we'll, get a better understanding of the brain, and have those type of biomarkers that could be neuronal signals that would then allow us to describe the abnormalities in all those patients more precisely. And one could definitely be related to different, sensibility to sensory inputs. We know, for example, that some patients with autism have what is called hyperacusis, so that they react differently to sound. They found also quickly that sounds are too loud, but then when we do a classical auditory test, they hear perfectly well. So it's difficult to quantify. So, hopefully, the more we understand the brain, the better we'll be able to actually classify, and then to have a more targeted treatment, or yeah, treatment, whether it's medical or therapeutic, to actually help those people or to understand them, better. So I fully agree with you that we are far from from uh, from where we should be. But hopefully, brain understanding the brain better will help us to address this. And again, like a little like what we call surprise, we shouldn't put everything under the same type of surprise. There's so many different types of surprises, and maybe looking more into this will allow us to know what brain circuits are related to each of them. Similarly, if we can dissect the different aspects of or symptoms that some autistic people have, maybe we can then also better understand what their condition is like.
Beautiful. Thank you very much. Yeah, because I was just asking the question because I was considering some people that might be told that they have autism, but might have a different perceptive
Yeah,
experience with the auditory processing.
yeah. I fully agree with you. I also know a person who was diagnosed with autism and then a few years later not anymore. And I mean, I'm not blaming for the clinicians. It must be extremely difficult to label a person. I mean, some cases are clear, but some others are. It must be extremely difficult because there's no convincing objective measure of those conditions for now. So I think that's really one of the goals of the research, is to try to quantify it in a way or another. Now, maybe this surprise response is something that could be used for the schizophrenia. For autism, it's some studies show it, some not. So maybe it's also because we put too many conditions in autism, and it should be seen as different disorders. And I wouldn't be surprised that in the future it will be called different. I mean, already 20 years ago we called it just autism. Now at least we call it autism spectrum disorder. So it's a little better, but I wouldn't be surprised that maybe some of them are just hyper perception could be something or different
You,
conditions for sure.
usually there are schizophrenia and autism depending on what level you are. These are comorbidities, and usually there is a misdiagnosis. And
Yeah.
also there are some people who are on that level and are considered having, some special kind of condition, and these are um, twice exceptional. Of course, some people are considered gifted in that sense. And one of the intersects that they have is just like when we go back to the original question earlier, was about, for instance, sensitivity to sound. And if we know the theory of the Brewskis over excitabilities, we could see individuals who are sensitive to their senses, like the sense of touch, the sense of hearing, for instance.
Mhm.
So this has a great intersect among gifted individuals and also people who are on the autistic spectrum. So the way we perceive the world, or they perceive the world, depends upon how they are having this kind of excitability. So it's, I think, quite difficult to really make, a final say on this, but these are the
Yeah.
things that should be considered. Yeah, sorry, there was someone who came on stage, who wants to come on stage. I hope I'm able to bring him. Ben? Ben, are
Ben's
you
on
here?
stage.
Oh, hello. Yeah, I just had a question, so I was coming up. I don't want to interrupt if you guys are in the middle of a presentation or something.
Yeah, there's a comment by Lubna on the room chat. We will read that after you. So please ask your question.
Um, so I gathered that you're talking about autism
Yes.
and ways of identifying it. And recently, I've been kind of seeing the hypothesis that it has to do with synaptic pruning.
Mm. Yeah.
That a lot of these types of things, that there's like an interruption in the normal synaptic pruning. Am I correct
Mhm.
in that?
Yeah, that's one of the hypotheses. Yes.
Okay, so I'm curious, is there just not like a brain scan you can do to be able to detect if synaptic pruning, you know, is on course? Is that
Yeah.
something? Go ahead.
Yeah, I sadly don't think that's the case. So I worked on something similar for my PhD study. It was a rat model of autism, and we did indeed find more connectivity in some types of connection. So the reason could be a lack of pruning. I think it's actually quite difficult to quantify. If it was abrupt, we would see it. If it was abrupt, the symptoms of autism would also be much clear to distinguish. I think actually we cannot really quantify, and for sure not in humans, if there's just a little change in pruning. Sadly, not not what I know of, and not because information still flows through. Um, you know, people with autism still hear, they can still see and everything. If it was drastic to the point that we could actually measure it non-invasively or even invasively, then then it would be much more drastic changes. So for now, with the technologies available nowadays, to my knowledge, there's no way of actually quantifying what should be quantified also. Plus, I have to come back to this aspect of autism, that it could be that for some people, it is a lack of synaptic pruning, and that's why we also think it's taking place in development. But for some other patients, it might be another reason. So also when we do studies in humans, it's also difficult because of lack of better definition, we actually group all those people, and then it might be difficult to diagnose it based on this.
Interesting. Thank you.
Mm. It would
Yeah,
actually
thank
be a tool that would also be interesting in for aging. Often the symptoms that come with dementia actually come much later than what the brain changes. So if somehow we could also diagnose some changes like pruning or loss of connections in the brain in a better way, I think it would help for a lot of diagnosis and early interventions that we know are better than later interventions. But we have what we have nowadays, so
Well, actually,
hopefully that will
some of the
change.
work that I had done in the lab was trying to develop therapies for like sensory drift and other things that are cognitive decline, and it was surprising how a small amount of almost gamification to the point of like 20 minutes a day was enough to sometimes stave off the sands of time, which is kind of surprising if you think about aging and how much more old people there's going to be going forward. So
Mm.
this might become a big problem going forward.
Yeah, yeah, for sure. Yeah, yeah. Well,
yeah.
there's still a
So
lot of things to discover in the brain, and that could be extremely powerful for so many different conditions. But
so
it's complex, so step by step.
Bob, there's a question I think here on
Sure
the room
thing.
chat. Can you please read it? And I would like to ask Tanya at this point. We've been here for 1 hour and 42 minutes. How much more time do you have for us?
Yeah, this is super interesting. I can definitely stay until 6 p.m. my time. I don't know where you are. Like for sure, 20 more minutes.
All right. So, yeah, there is a comment of Lubna here on the room chat, and I would like to request Bob to kindly read it.
Sure
Bob,
thing.
mic to you.
Uh, at what level can we use the biomarkers, cellular or molecular?
I mean, anything would be useful. So the suppressed response is not cellular or molecular, it's actually an electrical response. So anything that's an objective measure. Most of them are in the blood, and that's molecular, I guess. But uh, anything that would serve this again, this mismatch negativity response is not related to a cell or to a molecule. It's related to a whole network response. It's an electrical signal that we cannot get back to one particular circuit in humans, but doesn't matter. If it's a biomarker, if it's an objective measure, then it's good. As long as it's not subjective, as soon as there's some subjectivity, then it's very, if the patient, for example, has to report, oh, I feel like that, or I see that, or I hear this, then it becomes subjective, and then it's very difficult. As long as it's objective, then I think we can use it.
So Lubna has a secondary question. I have read this morning a paper relating to some microbe infection in the brain with Alzheimer's. Are
Mm.
microbes considered in diagnosis in disease related to the brain?
Um, yeah, thank you for the question. I'm afraid I'm not a good person to address this because I don't, I don't know that field. I would guess that in this case, for example, the microbes actually change something to the way neurons work, or
Well,
to the
usually the
way they,
toxins produced by the byproducts of
yeah.
the pathogens in like metabolism or whatnot, there's usually some sort of molecule there that's problematic for humans that usually creates a cascade effect. But it would be interesting to hear about the paper. Please do post a link to the paper. We'll definitely take a look. But yeah, that's a very exciting direction as well.
Mm.
Yeah, so thanks. And yeah, since we have a little more time, I would like to read a question from uh, someone who had sent it to me. From Mariana. How might the delayed maturation of the cerebral cortex impact cognitive abilities and emotional regulation during adolescence and early adulthood?
Yeah, I think that's very interesting, and we can look at this later plasticity as both an advantage because that allows the brain to really shape to many things and to learn and to change and to improve. But it can also be, it's also a time of vulnerability in a way, so where the brain can change, but in the not in the way we would like it to change. So yeah, it's a time when the brain has a lot of potential and it has a lot of vulnerability also. Um, I'm not sure. So, the question also relates to the emotional changes happening during adolescence. So I think that's slightly a different question because, again, I try to look just at very sensory processing. Of course, there's a lot of emotional changes taking place also during these times. That's probably related to other things, but that also influences whatever is going on at the sensory processing. So I think they're both related, and and yeah, I think both aspects. I think this plasticity is fantastic because it allows the brain to really adapt and to learn and to change. But it's also a vulnerable period, so where we have to be careful. For example, if the brain, in that case, in the case we tried in the lab, is was the environment was modified, was masked, and then somehow the mice didn't respond properly to surprise. It's like if they were surprised for everything, unlike the control mates that had not been exposed to an abnormal environment. So it's definitely something that we have to consider. And I mean, I think intuitively we know, when we say that um, children and adolescent brain are more vulnerable, for example, to drugs or loud sounds, all this, that's correct. And I think that has to be seen with those two aspects of both a huge potential, but also vulnerability. I'm not sure if that really addressed the question, but
Yeah, thank you. And there's another question here by Bing Baez. She said, considering the role of surprise in learning and curiosity, how might educators harness this knowledge to enhance
yeah,
student engagement and retention?
yeah. Yeah, I think that's also a very interesting thing to keep in mind. So when I consider when I was at school or when my kids now at school 20 or like 30 years later, the way that people teach, I think, is very different. When now it seems like engagement is much more important. When I was at school, we were just sitting on our desk and listening to whatever the professor was saying. Where I think now engagement and active participation is much more put forward, at least in Swiss schools, and I think that really engages, not only hands-on experience, but also surprise or everything that's more active and not just passive. And I think that really triggers, increases learning and motivation and all this. So it's partly related to surprise, I would say, to the role that surprise could play in learning, but also to many other aspects like engagement, alertness, as someone mentioned before, excitement also. And I think that's definitely important for learning. Um, yeah, I
Yeah,
don't think.
thank
I mean, surprise has many aspects. Also, if it's a fear surprise, then it's definitely not good for learning. But or it's good for one type of learning, but maybe not the one we would like kids to have at school. So yeah,
Yeah, thanks. So I think this will be the last question from Paul
yeah,
George. He said, "Are there any parallels between mechanisms observed in surprise processing and those implicated in other cognitive processes, such as memory formation or decision making?"
yeah. So we haven't. So some people have looked at the role of surprise with memory formation, and they showed a directly link between a computational role of surprise uh, for memory formation. Some have also shown a direct link between surprise and memory segmentation. Um, I guess this is related more to categorization or things like that. So there's definitely some studies that show the link between surprise and memory. And for this, I can just refer to papers. And I think an interesting paper that summarizes this is one by a professor of computational neuroscience called Wolfram Gerstner, and it's called A Taxonomy of Surprises. And there's some, I think, a very nice summary of the different aspects of surprise, but also of all the papers that have been looking at how surprise affects, for example, memory formation. There was another part of the question I forgot now.
Ah, yeah. So he was asking about memory formation or decision
Decision
making.
making. Yeah, so decision making would, I mean, of course, it depends on memory, but I think decision making relates a lot to predictive processing in a way, and surprise is definitely related to this. So, um, again, predictive processing, it compares what you expect the world to be with the sensory input. And if there's a big a match between those two, then I think that, so no surprise, then I think the decision is easy to take, because then whatever you expect is what you have, and then you decide in an easy way. Now, if it's not what you expected, so if you have a surprise response, then I think this definitely influences a decision. For example, if you, now how we do it with mice is, it's if they hear a frequency, they have to lick on the right side. If they hear another frequency, they have to lick on the left side. So if they expect a frequency to go on the left side, uh, and they hear that frequency, then they will decide quickly to go on the left side. If now they hear a frequency that doesn't fit with what they expected, then they will be unsure about what to decide, and then the decision will first be less precise and will be later also. So I guess, yes, surprise definitely affects decision making in the context of this predictive processing framework.
Yeah, thank you, Tanya. So there's a comment here on the room chat by Lubna. I think she wanted to put a link here of the paper she mentioned, but she could not find it. Lubna, please message me or Eric, uh, or IO. Private message me on that link, because we don't have a portion here in Clubhouse anymore where we can put the link. So yeah, at this point, we are near the top of the hour, and I would like to thank our special guest speaker, and Tanya, and uh, we hope that we can invite you again here in Quantum Photonics. It was a great listening to your talk, and thank you so much for the great conversation. So I'd like to pass the mic to you. Tanya, do you have some comments or parting shots?
So yeah, first I would like to thank you, Cecile, for this opportunity. I think it's very nice for me and for science in general to be explained, to be allowed to explain the science, and also to a wider audience. And maybe to the audience, I would say thank you for all your super interesting questions. And I think surprise in general and the brain development is also, we're just scratching the surface of it, and I think there's so many different directions we still have to go, and so many unknown, and some of your questions really raised interesting future direction of research we can go to. So stay tuned, and yes, I hope that with time we'll understand better how surprise changes during development, and how this is related to all the different aspects you raised in your questions.
Yes. So everyone, let's flash our mic for our special guest speaker. Thank you so much, Tanya. So we were like clapping for you when we flashed our mic.
Yeah.
And I'd like to ask Bob to say something if you have something to say before we give the mic to Darcel to help us close the room.
Thank you, Cecile, and I wish to thank our wonderful speaker on behalf of everybody in the audience. I think we all had a spectacular discussion today. Thank you very much.
All right, so we come to the end of this session. Thank you, Ayo, Kevin, Kyle, Ben, Giri, and Darcel is here to help us close the room. I cannot say all of your names here in the audience, but as usual, thank you so much for your support. So, yeah, Darcel, I'd like to pass the mic to you now.
Thank you, Cecile, and thank you, Doctor, for a great room. To close the room, we're going to start our countdown from lucky number 9. Is everybody ready?
Ready?
9.
9,
9,
Gary, 8.
8,
Ben, 7.
7.
Kyle, 6.
6,
Size.
Kevin, 5.
Quattro.
Io, 4.
IO 4.
Bob, 3.
3 it is.
Cecile, dos equis.
Dos Equis.
Good doctor, your numero uno. Call out our last number, please.
1.
Yeah, thank you, Dr. Barkat. See you back in quantum photonics soon. We had fun time with you. Superb. So good night. Goodbye, everybody. See you on the next rooms.
Goodbye. Thanks a lot.
Take care.